Let's talk about vaccination. Vaccination is all about inducing an active immune response, either humoral, cellular, or both, against specific pathogens. Now, there are different types of vaccines. 1. Live attenuated vaccines. These are made from microorganisms that have been rendered non-pathogenic, but they still retain the ability for transient growth inside the host once inoculated. For example, some live vaccines like MMR, measles, mumps, rubella, and varicella can even be given to people living with HIV, but only if their CD4 plus cell count is above 200 cells per mm superscript 3 and after consultation with an infectious disease or immunology specialist. The pros. Live attenuated vaccines induce both cellular and humoral responses, and the immunity is usually strong and lifelong. The cons. The microorganism can revert to a virulent form, so these vaccines are contraindicated in pregnancy and in patients with immunodeficiency. Examples include adenovirus, non-attenuated, given to military recruits, oral typhoid, Ty21A, oral polio, Sabin, varicella chickenpox, smallpox, BCG, yellow fever, intranasal influenza, MMR, and rotavirus. Here's a mnemonic. Attention teachers, please vaccinate small, beautiful young infants with MMR routinely. 2. Killed or inactivated vaccines. These are made by inactivating the pathogen with heat or chemicals, while carefully preserving the epitope structure on surface antigens, since that's what the immune system recognizes. They mainly induce a humoral response. The pros, they're safer than live vaccines. The cons, the immune response is weaker, mainly humoral with little cell-mediated immunity, so booster shots are usually required. Examples include hepatitis A, intramuscular typhoid, bipolysaccharide, rabies, intramuscular influenza, and polio, Salk inactivated. Mnemonic, a trip could kill you, standing for A equals hepatitis A, T equals typhoid, R equals rabies, I equals influenza, P equals polio, Salk. 3. Subunit, recombinant, polysaccharide, and conjugate vaccines. These use specific antigens that are most effective at stimulating the immune system. Polysaccharides can be combined with conjugate proteins. Pros, they target specific epitopes, so there's a lower risk of adverse reactions. Cons, they can be expensive, and the immune response is often weaker compared to live vaccines. Examples include hepatitis B, HBV, antigen equals HBsAg, human papillomavirus, HPV, acellular pertussis, AP, Neisseria meningitidis vaccines, various strains, Streptococcus pneumoniae vaccines like PPSV23, which is primarily T-cell independent, and PCV13, PCV15, or PCV20, which produce a T-cell dependent response. Others include Haemophilus influenza type B, HIB, and herpes zoster vaccine. 4. Toxoid vaccines. These are made from denatured bacterial toxins, but with the receptor binding site kept intact. This way, the immune system can generate antibodies against the toxin without any risk of causing the disease itself. Pros, they provide protection against bacterial toxins. Cons, the antitoxin antibody levels decrease over time, so booster shots may be needed. Examples: Clostridium tetani, tetanus, and Corynebacterium diphtheriae, diphtheria. 5. mRNA vaccines. Here, a lipid nanoparticle delivers the mRNA into cells. The cells then use this mRNA to synthesize a foreign protein. For example, the spike protein of SARS-CoV-2. Pros, they have high efficacy, induce both cellular and humoral immunity, and are safe in pregnancy. Cons, they often cause local and transient systemic reactions such as fatigue, headache, and muscle aches. Rarely, they can cause myocarditis and pericarditis, especially in young males. Example: SARS-CoV-2 mRNA vaccines.